analysis. So for network pharmacology, we have discussed about what is system pharmacology and what is network pharmacology. I told you a collective, the network pharmacology is, of course, a part of systems pharmacology. Other than that, we also study about drug protein interactions, pharmacokinetic compartment models, pharmacodynamic models, all these things we observe in systems pharmacology. Okay, so that's what I explained on yesterday. And then I came to the pathway analysis. For that, I have shown different interaction networks. I have taken you to the string DB and I have taken you to the fun coop. Okay, so these are some of the databases we have seen uh, to download the networks. Okay, so if you want to download the networks, okay, it can be funcoop or string DB. What is the file format we can download from the database? My first question is, what is the file format of protein interaction networks? Very good. The answer is TSV, tabulated separated value file. Okay, so this you have to download, and if you want to edit something, you can edit through Excel, and if you want to go for display change of display in the interactions, you can go for cytoscape network visualizer. Okay, so this is how we have seen, and we have also seen a few plugins. There are plenty of plugins. uh, Whether I have shown you few results or not, I don't know. No. So you remember what plugins I have shown? I have shown you something related to. So whatever plugin, mention me any two plugin examples for cytoscape visualizers. Mention any two plugins. I'm not asking even applications. If you know two plugins, you can mention. Funcoop is a database. If I ask you about some two plugins, there are so many options. Okay, Cytoscape is of course like a package. In that you can install some plugins or some softwares. Okay, so which you can work. Now I can show you later to that. Uh, so yesterday we discussed about Cytoscape, isn't it? So the continuation. So if you see the Cytoscape ecosystem, okay, we can able to generate the models, okay, and we can use plugins like Plugo, SC NetBridge. Okay, there are so many plugins available. That is, after installation of Cytoscape of recent version, even if you want to download the networks, if you want to visualize the networks directly from the string database, it is possible with you. So you can directly export the networks in the Cytoscape visualizer by clicking a hyperlink in the string database. And similarly, you can integrate with these programming languages, that is Python. Okay, I think Python is generally used for developing artificial intelligence applications, isn't it? If you want to develop your own applications, so we rely on the Python. So it's also integrated with this programming language. And if you want to draw some kind of a graphs, plots, etc., even you are associating with R scripts. Okay, so even it supports the R programming language. So from this, you can able to understand. So of course, this is a foreground, but also in the background, we can use a database like string, and even even in the funcoop also, you can directly upload the structures. Okay, so whatever you upload, okay, so you can also integrate with these languages, and you can change the interactions. So you change the color, change the display style of the interactions of the node interaction. Not only can change the nodes, even the edges also can change, like color, thickness of the line, etc. All these things you can make it out. So if you see the compound interactions in Stitch, you remember yesterday I showed you directly exporting from the website string database. So if you see the cytoscape network, okay, so we have an option there, okay, to just create a kind of a network. If you search for one particular gene, now I just search for a gene like TRAP1. So for this, there is no need to have any plugin. Okay, simply have to, if you want to develop a network, okay, which you can use it for your publication, you can open it in Cytoscape, okay, and then you can have to simply build a network. It's simply a single click button. You not only get this kind of interactions, okay, like this, you also get the rankings. If you want to find the top 10 genes, okay, based on the interactions, which is most close, if you see the trap one, the most associated protein, linked protein is HSPA9. Okay, so this is also shown in colors. Okay, the histogram of colors. The red is more associated, and if you go to the yellow, you find it is less associated. But out of plenty of nearly 23 proteins, how many? 1, 2, 3, 4, 5, 5, 25, 28 proteins. Out of 28 proteins, you can find 10 important proteins. Okay, so this is a kind of a gene enrichment you can even do using the cytoscape. And using the CytoHubba plugin, you can also get this information related to betweenness, closeness, which is closely linked with the protein that you get a high degree. HSPA9 is going to have a high degree. Not only that, you also have betweenness, closeness, high values. Okay. So we try to also find like that. And if you want to go for network analyzer, you have to install this plugin. Okay. And Clugo, there is another plugin where you can link the different pathways. Okay. You can link two or three pathways and you can find which are the most important genes. Okay. In that interaction pathways. Okay. This you can find from the Clugo plugin in Cytoscape. Okay. I believe I sent you the DOI of the latest published articles in system biology, system pharmacology. You remember yesterday before closing, I just sent you the DOI. Okay, so you can use it for downloading these open access publications. Of course, each and every publication expense is around more than 2 lakhs. Okay, of course, just because it's published in open access, any people can access it by using the DOI. And if you see this Krugo plugin, you can also get the information, okay, in the form of a pie chart. Okay, there are different pie charts also you can get from this Krugo plugin. And clustering coefficient, this is an additional information you can generate. Node degree distribution, path length distribution, and there is also one more plugin called M code analysis. Okay, this M code analysis is used to recognize the most important modules like if you want to find which are the key regulatory proteins, associated proteins. So all these things we can able to differentiate using this plugin. You can change the networks, you can change the display style however we want. Okay, so we can also get even the scores for the interactions. So this is something related to the application of plugins. Okay, so I think all these things I have shown, like how to open a network in the fund group and how to filter it based on the pathways or based on the specific organs. Okay, so some information is shown. Okay, just I recap you. What are the different plugins we can use in pathway analysis? Okay, now let me go to the today's new chapter. That is the 10th chapter on concepts of immunoinformatics and reverse vaccinology. If you see the our certificate course contents, okay, so the we have completed target discovery, drug design, target discovery for first 2 days, drug design for the next 5 days, system biology approach for the eighth and ninth session. Now for the three sessions, we'll be discussing on vaccine design. So the basics of vaccine design were related to reverse vaccinology and what are the different immunological databases we use. Till now, whatever databases we have seen, a common biology databases, biological databases, or common oncology databases. Other than these databases, we use exclusive databases to study immunity. Okay, that's what we call that as an immunological database. We see something new in this session. Okay, after that, we discussed specifically on T cell epitope and B cell epitope vaccine design. Okay, now let me go inside this topics. Immunoinformatics. What do you mean by immunoinformatics? We have heard about bioinformatics. Okay, and also we heard about different omics, isn't it? Genomics, proteomics, intraomics, transcriptomics, metabolomics. So many omics are there, okay, based on what exactly we study. But what is this immunoinformatics? So this also called as computational immunology. Okay, this is a separate field of science that encompasses high throughput genomic and bioinformatics approaches to immunology. So if you come across this computational immunology, here I basically going to explain you how to design vaccine. Okay, so here majorly I focus on designing the peptide-based vaccine. Okay, so how to design a peptide-based vaccine, and that too, whatever vaccine we are going to design, this is targeting very precisely to the target population. For example, if I want to prepare a vaccine, okay, for Tamil Nadu population, especially even in Tamil Nadu, if I am concerning on Nadar community, okay, that is possible with me to design precisely a vaccine for the Nadar community. That is whatever I'm going to design, it is going to target 99 percentage. It's going to have 99 percentage or more than 99 percentage efficacy to that community people. You got it? So not only the drugs has to be designed precisely, even the vaccines can also be designed more precisely. Okay, so for this, we should have this kind of immunoinformatics or computational immunology knowledge, because we are basically even touching the HLA alleles. You know, each and every person, even if you see in the siblings, okay, sisters born at the same time or brothers born at the same time. So if you see them, okay, so their genes are most similar, isn't it? So only they are getting more similar phenotype. They look very similar, okay, in everything. Can be skin color or can be hair color or how they look, okay, everything is more similar. But even the siblings vary with respect to immune proteins, especially HLA allele proteins. Okay, so I mean, they vary it like how each and every person has a unique fingerprint. Okay, likewise, even the immune proteins. Okay, if you see the HLA alleles vary even with respect to the siblings. Okay, so one should able to understand. Okay, so what kind of alleles are frequently transferring during the heredity. If you see the community, Nadar community, most of the people who's going to have a similar kind of an alleles transferred from in generations, isn't it? They are transferring from one generation to the another generations. So if so, it is easier for us if you target these kind of alleles, HLA alleles. Okay, more about that we'll be seeing in the subsequent slides. Okay, if you see this kind of immunoinformatics, okay, we basically use to convert the immunological data into computational problems. This is one such example I narrated. Okay, like targeting, designing a vaccine targeting for even up to the community people, not even up to the country region or even the state region. Solve these problems using mathematical and computational approaches and then convert these results into immunologically meaningful interpretations. Here, a lot of tools available in a single web resource. Not only you find plenty of data deposited in the database, you also have plenty of tools, okay, in the public domain database, and also you have other exclusive tools present in various website. Okay, so you can able to download all this information. Okay, but how to interpret is the most challenging problem. Okay, so you get results, but one should aware of how you have to infer the results. Okay, So for this, of course, I think this certificate course will give you some kind of ideas to. Now, let me go to the next point. After the recent advances in sequencing and proteomics technology, there have been many fold increase of molecular and immunological data. Okay, if you see this kind of inference or interpretations, what I mentioned in second line, you try to get multiple scores. There are so many scores generated from the software, but you should know what is the exact formula or a correlation exactly used to calculate the score values. Okay, so one should understand the formulas in the scores generated from the various tools. So that's the most challenging, of course, for interpretations. And if you see here, most of the recent technologies are used to develop these kind of a data deposited in the database. So of course, we are going to take even recent updated data from genomes and proteomes derived from the humans and other animals also. Not only you can design a vaccine for the humans, even veterinarians, if they want to design a vaccine for cattle or for a pig, sheep, not only these domesticated animals, even they can go for any even few wild species also. Even for camels, you can able to bring a vaccine. Okay, so let me come to the definition of vaccine. What is a vaccine? How a vaccine can differ from a drug? A vaccine is a biological preparation that induces acquired immunity against a particular pathogen. So when we go for designing a vaccine, even for SARS-CoV-2, I think everyone have taken the vaccines like COVID, the Covaxin or the Covishield, isn't it? So what is going to happen? So whatever Covaxin or Covishield you take, that is going to give you an acquired immunity. Okay, so whatever antigens we take, these antigens will create antibodies which can combat that specific strain of the pathogen, isn't it? So that is a definition for vaccine. So it doesn't limit up to the targeting the pathogen. Even we have vaccines for the treatment of cancer. Okay, so if you want to get an vaccine, okay, so you take a kind of a target the cancer, you put develop an antigen, try to develop antibodies from that, or even we have so many antibodies directly developed to target the specific cancer. Okay, so there are different ways of treatment of cancer. Either you can use the antibodies. If you use the antibody, okay, that gives a remedy for shorter period because of humoral immunity generally prolongs immunity for very shorter periods. But if you go for an antigen, then obviously the patient survival rate will be increased. But taking the antigen vaccines are very painful. Okay, if a patient is in third stage cancer or he surpasses the third stage. Okay, if there are five stages of cancer, okay, and if a patient is in third stage or is crossing the third stage, then obviously taking an antigen is not wise. Okay, the doctors won't prescribe, the oncologists won't prescribe to take the antigen. Instead, if they want to survive for a shorter periods and if they want to relieve pain during the survival, the doctors prescribe to give antibodies. Got it? So we don't have a permanent remedy for the treatment of cancer. Just for increasing, like if a patient is mentioned to live for 2 days, okay, but if you take the antibody, if the patient is given antibodies, then he can live 3 or 4 days, not more than that. Okay, just for increasing the shorter span life. But antigens, obviously, for a longer periods. And if you see the vaccine that requires adjuvant, okay, to stimulate an effective immune response. The adjuvant can be a plant-based or biological-based. Okay, it can be of any source. Okay, even myself, I am working on adjuvants for past 13 years. I am basically working on plant-based adjuvants. Okay. So not only I'm working on a plant-based adjuvants, but also working on few antibodies and few cells, the immune cells, which can also work like adjuvants. I basically worked on dendritic cells. Even not only the antibodies I used, like IL-4, GMCSF, IL-2. Okay, these interleukins and the growth factors are also considered as adjuvants. Other than that, even whatever dendritic cells I cultured, that itself will be acting like an adjuvant for another cell. Okay, so I worked on almost all the antibodies, plant-based adjuvant cells, etc. Okay, to develop a vaccine. So if you see the vaccine properties of vaccine adjuvants, or whatever antigen you use to develop a vaccine should be more immunogenic. Okay, we also have a tool to calculate the immunogenic properties through immunogenicity score. Then one should know what is the correlation used to calculate the immunogenicity score. Okay, not just inferring the result whether it's a maximum or minimum. You should also have a knowledge on the scores whatever they generate in the we find generated in the tool. And if you see the a second important property is the high antibody production. So whatever vaccine adjuvants, if you think if you use a plant-based adjuvants, like generally the most widely used plant-based adjuvant is the saffronin. Okay, and most widely used chemical is the alum. The alum is used for the past 120 years. Okay, so for the past 12 decades, okay, most of the vaccines, just because of low price, alum is used as the universal adjuvant for most of the vaccines. Okay, but alum is a metal oxide. Okay, so it's a metal compound, isn't it? So if you want to avoid this kind of an aluminum salts, okay, obviously one can also go for organic kind of a adjuvant. So and I am using the plant-based organic compounds. Okay, so you don't going to accumulate in your cells. Okay, so these are the recent researches happening in the world. And if you see the third important application, so high antibody, of course, these high antibodies are produced from the B cells. Okay, B cells are produces high antibodies. So the for activating the B cell, of course, you need a adjuvant. Okay, you of course you need a vaccine, but adjuvants increase the activation, so you get high antibody production. Not only that, you can also generate specific cytotoxic T cells. The cytotoxic T cells directly combat the cancer just like antibodies. Okay, you don't require any other cytokines. Even the T cells, cytotoxic T cells itself go and combat, and even the cytokines released from the cells can be helper T cells and cytotoxic T cells. Again, will try to combat the cancer cells. Okay, so both proteins and cells together fight. Okay, with the cancer cells. And if you see the types of vaccines, there are plenty of vaccines. The major classification of vaccines are inactivated vaccines, live attenuated vaccines, messenger RNA vaccines. I think you heard about these vaccines during the treatment of COVID-19. Okay, mRNA vaccines, viral vector vaccines, Covishield, and the previous one is the Covaxin. So these vaccines play important role to relieve from this COVID-19 infection, isn't it? To relieve from SARS-CoV-2 infection, isn't it? So likewise, you can also see subunit recombinant polysaccharide conjugate vaccine. Like if I like to work on liver cancer, okay, one should also think about to bring the vaccines in the form of subunit recombinant polysaccharide conjugate, or even you can go for inactivated vaccines. Okay, so these are the two types of major vaccines used for developing a vaccine for liver. Okay, because of hepatitis infections. And if you see the toxoid vaccines, okay, can used for to treat diphtheria, tetanus, And peptide-based vaccines, the popular one is the Covax. So we just hearing about only the Covaxin and Covishield, isn't it? You know the efficacy of the Covishield. So actually, if you see the SARS-CoV-2, okay, we have hundreds of strains. Initially, whatever Covishield and Covaxin developed is going to target only the initial strain. Okay, the first strain in 2019. But when our country is declared as a pandemic, okay, in March 2019, okay, we don't have any approved drugs or vaccines for treatment. So the doctors prescribed only the repurposing drugs. The drugs is for malaria, chikungunya, dengue, okay, influenza. All these things are prescribed in the A drugs prescription, along with the anti-HIV drugs. I think I mentioned you this in earlier class sessions. So if so, okay, if the how come a patient can be treated with the repurposing drugs, which is not going to target the major spike target protein of SARS-CoV-2. Okay, for dengue, malaria, we have different kind of proteins. We have a proteases or polymerases as a targets. But how come the main target proteins are spike glycoprotein can be targeted using these not insignificant drugs? So one should think about the major target, then only we can discover not only a drug, even the vaccines. Okay, if you see the kind of mRNA vaccine or this kind of viral vector vaccines, they have developed only from the spike target receptor. Okay, so even if you see this vaccines, I just ask you a question. Okay, so my first question is like, what is the efficacy, because everyone have taken Covishield and Covaxin, either Covishield or Covaxin. Can someone tell me what is the efficacy of Covishield and Covaxin? Okay, uh, in 2020, that is for the initial strain of SARS-CoV-2. But 2021-22, you can find more resistance came, and even the efficacy is decreased, obviously. And even because of more strains, whatever vaccines we developed, especially if you go for Delta, SARS-CoV-2, Delta, Epsilon, a series of strains are there, isn't it? Alpha, you have nearly 6 mutations. Beta, you have 8 to 9 mutations. Gamma, Delta, so many kind of strains are there. But for the initial strain, okay, during when the 2020 we got the vaccines from Government of India. Okay, so what is the efficacy we observed? Efficacy here I mention about how much efficient your vaccine has targeted. Some kind of a random number you can tell. What is the efficacy percentage for the Covishield? So if you see Covishield or Covaxin, good, you have tried very good. But actually, there are two boosters. If I take first booster, I get nearly 56 percentage, not more than that. 54 to 56. But if you go for the second booster, obviously it increases the dose response. I mean, the adjuvant vaccine response. If so, it came maximum of 84% for the Covishield. Good try from everyone. 84% in 2020, but in 2021, obviously it reduced. Okay, 70 something. Okay, so for if the efficacy is 84 percentage, at that time when they measured the Covaxin, it was 91 percentage. But if you see if, for the Delta strain, I think Covishield comes around 17 percentage efficacy, 1 7. So you understand, only 17 out of 100 patients will be not getting that kind of infection. So it varies. Okay, so whatever vaccines they developed, okay, it won't work for all the strains of SARS-CoV-2. So this is the exact percentage, okay, for the initial strain. But the Covax, whatever I've shown, okay, this is the polypeptide vaccine, which is which was discovered in USA. So other than these two vaccines, we also had 87 vaccines in 2020. In I think September or October 2020, all over the world. Okay, nearly 89 vaccines came for play. So if you see this COVAX, okay, which was developed in USA, most of the vaccines are from USA. I think more than 40 to 50 vaccines are developed from USA. Okay, this is one such vaccine, COVAX. This is a multi-dope vaccine. This yielded more than 99% efficacy. So most of the patients, those who are taking this, uh, most of the patients are not getting any infection if they take the COVAX. Okay, so why I'm mentioning specifically on the COVAX, because in the next two sessions also I'll be discussing you about how to go for designing these kind of peptide-based vaccines. Okay, so now let me take you to the concept reverse vaccinology. In immunoinformatics, one of the major area to work on vaccine design is reverse vaccinology. So before going to the reverse vaccinology, one should understand what is classical vaccinology. Okay, so I think everyone know that the biotechnology gets into popularity only because of vaccines. So even the origin of biotech, we can tell, or the origin of vaccines and the origin of biomolecules like DNA, RNA, etc. Isn't it? So vaccines is most important. So how these vaccines are discovered? For now, most of the people don't have the polio infections, don't have the smallpox. Almost it is curbed. Okay, this is actually totally curbed because of the vaccines taken during the childhood. Okay, so how these vaccines are discovered initially? So there are so many protocols, so many informations, but generally, if you see these vaccines, okay, these are developed from the dead viruses, okay, or there can be inactivated virus. We heat, we give a kind of a treatment in such a way that the virus is going to lose its pathogenicity. If virus is going to lose the pathogenicity, obviously this cannot be used for the treatment of that kind of viral infections. Okay, only one type of virus is going to kill that one type of virus. Okay, can't use something like the hepatitis B treating hepatitis C. Okay, so not only we take the live, I mean, attenuated or killed care of viruses, but you can also take the particles, the proteins, okay, the peptides derived from the viral particles for the treatment. So, whatever we see in market, okay, whatever we see the vaccines in the market are produced in large scale through the fermenters. So the bioreactors, they produce large quantity, and of course they try to we are trying to use these kind of vaccines, okay, based on the production. If you see the production of Covishield and Covaxin, think about that when they have started the production, when they have completed. So within a very short period of time, okay, so eventually they come up with large numbers, okay. So one should aware of even the type of vaccines, okay, how they can derive, whether the proteins are used for treatment or mRNA derived from the virus is used, or even you're taking a portion of the particles, okay, or you're taking a proteins, peptides, everything concerns on development of vaccine. That's what we call it as a classical vaccinology. But reverse vaccinology, okay, we are not directly taking from the viral particles, not taking from the bacteria or parasite particles, but we start digging, okay, from the already existing genome data and proteome data. Okay, so only in 2020, if you see the 2003, the final human genome was drafted, but it started in 2001, but they drafted finally in 2003, and rat mice genomes drafted in 2002, 2001, like that. Okay, so most of the genomes, so animal genomes, are drafted during 2000 to 2005. If so, think about the proteomes. One day after the emerging of genomes, Okay, then only even the proteomes, the protein-protein interactions, and other kind of information related to the proteins, okay, or discovered. That is after 2005. So from 2005 to some nearly 2013, something like that, you find numerous proteomes are deposited in the database. So from whatever data is deposited from the bioinformatics approach, we use this kind of forms, okay, to try bring a kind of a vaccine. Okay, so initially what we do, we take the you have any doubts? Okay. So you take from the major virus or bacteria and you bring the vaccines in classical. But in the reverse vaccinology, you're not taking the virus, but you're taking the already developed information, bioinformatics, biological data information, and you try to bring the targets and you try to get a kind of epitopes or any other antibodies kind of vaccines like that. Okay. We go from reverse. We're not trying from the base like virus bacteria, but we go from the reverse with the already developed data. If you see the reverse vaccinology, so here generally you take the genome sequence, you go for computational analysis, we bring the antigens, okay, we develop the antigen select. I mean, we bring the T cell and the T cell epitope based uh, peptides of after selecting the antigens, and then we go for checking the protective immunity, and we can also check the molecular epidemiology. This is most important. So we are not sure, like if you see the cancer, especially melanoma, you heard about that melanoma? What is melanoma? What type of cancer is melanoma? So this is my question now. What type of cancer is melanoma? It is a kind of a skin cancer. Okay. So actually, if you see this skin cancer, whether you find skin cancer in Indian patients, no, very, very less. If you see 0.0001 something like that, 1%, okay, that much only the Indian people can or succumb to that kind of skin cancer. Okay, you didn't hear many of the people is in India is facing this problem, but if you see in US, okay, because of that specific alleles, okay, you find more than a percentage. Okay, it's not even point percentage, even some 1 point something like that. Okay, so many people are more prone for the skin cancer because of that pale skin. Okay, so they have some kind of an alleles on that. So what do you understand from that? Even the cancer spread, okay, because of hereditary and other causes. So this also gives an important information to target vaccines, okay, based on the particular region. So the cancer vaccines uh, or the most developed vaccines, okay, of course in USA. So not I also mentioned you like if patients are suffering from a particular cancer in a particular region, okay, of a particular community, even we can able to bring a precision vaccine, isn't it? So of course one should aware of the molecular epidemiology of the spread of cancer. And of course, we have to concern whether whatever vaccine you design, it is non-pathogenic, it is non-toxic, non-virulent, of course. Okay, it is safe to use. So all these things has to be considered. So all these things, whatever I mentioned, can also be predicted from the tools, immunoinformatics tools. Okay. So everything you can able to find the information from the tools. Only thing is that you can try it out using in vitro or in vivo studies. Now let me go to a chapter called an example for reverse vaccinology. The first vaccine developed from the reverse vaccinology is meningococcus B vaccine. You know, meningococcus B vaccine is used for treating bacterial meningitis. Okay, so bacterial meningitis, of course, this kind of infection you can find mostly in the brain. Okay, so to treat this kind of a bacterial meningitis, okay, so participant, I think one is. Unmute. Who is that? I think he forgot to unmute. Okay, let me continue my session. So I was speaking something related to the example, okay, for reverse vaccinology, that is meningococcus B vaccine. Okay, so am I in the session? Am I audible to everyone?
Yes, sir.
Yeah, yeah. Okay. Because I don't know when my data is going to cease. So I just am checking. So thank you for your response. Okay, let me continue. Uh, if you see the reverse vaccinology, the first vaccine is meningococcus B vaccine for treatment of bacterial meningitis. Here they're using a series of protocols like virtual screening is also used here. Initially, 600 potential vaccine candidates are identified, okay, from the computational methods, from the sequences, from the target sequences of of the meningococcus. But if you see the kind of development, okay, so not just the vaccine candidates, whatever you generate from the tool and databases won't give you a kind of a positive result. One has to check, okay, that whether these proteins, this vaccine can be produced in real time or not. So the second screening they count for whether these proteins are successfully expressed in E. coli or not. Only 350 proteins are expressed out of 600. Later, the next ritual screening happened with to find how much surface exposed proteins are there. Okay, surface protein are important to target most of the surface of the viral target. So they identified only 91 out of 350, and only 28 novel proteins have shown the bactericidal activity on bacterial meningitis. Okay, so obviously if we try to use these proteins, from these proteins, they have come up with a kind of peptides. Okay, they designed the peptides, and these peptides have targeted more. So I think whatever vaccines developed for the meningitis is treating 99% and more. Okay, it's showing more than 99% efficacy. So this is a successful vaccine and first vaccine developed using the reverse vaccinology. And this is the article I published during the COVID-19 lockdown. If you, this is based on Nipah virus. Okay, I think everyone aware of the Nipah virus infections happened in 2018 in Kerala. I think even we have a movie, isn't it? Virus, something like that. A movie called Virus. So here, and so what do you understand from this these kind of infectious viruses? So this infectious virus spread, okay, if it happens in human and if it is not diagnosed, okay, so they are you can find 100% fatality. Okay. So during the spread, the doctors confused. They don't know what exact infection it was. But, Dr. Ashok Kumar, who is working on Nipah virus in USA, he came for vacation in Kerala. To see, he came luckily to that same region, and he was so interested to know about what's happening inside. And then he found the patients are infected with these kind of exact his research Nipah virus infections. Then obviously these people are isolated at the time itself. So the doctors made they take the patients and they isolated in the hill regions. They won't come down. They are isolated the hill regions and they as such. Okay, I think a lot of stories are there. So one should aware of any new viral infections, upcoming infections. So if any new viral infections come, okay, you can use this also as a template kind of a article. Okay, so a kind of a research. I just worked on that. So I just published only T cell epitope, but I worked on B cell epitope. But I have a lot of data. So just limited up to this. Okay, but exactly if you see the Nipah virus, the best vaccine for the Nipah virus is the B cell epitope. Okay, humoral immunity gives a better kind of successful results for this infection. Okay, but for few viruses, T cell epitope is the best one. Okay. Yeah. So if you see this of course an outcome of a funded project, but also these also awarded as one of the best project. Okay, during the national project grant. And if you see the workflow of the Nipah virus protein, okay, I think when I started the research, I found most of the structural and non-structural proteins are already worked by many scientists in the world. So the only protein they didn't touch is the protein W. So just I taken this protein for my research. But you see, if most of the viruses, whatever is there, okay, whatever is known, okay, for all the viruses already reverse vaccinology was already completed. If you get any new infection for even if you see the SARS-CoV-2, I was explaining you something about different strains and different mutations, isn't it? We don't have vaccine for all the strains and mutations, but if you find a significant population in the mutations, okay, or in those particular strains, then you can try to develop these kind of a methodology to develop the vaccines. Okay, let me go to that. I just taken the Nipah virus protein W and go for the antigenic protein identification. Of course, out of a series of proteins, I identified only one protein, and then I've gone for T cell epitope prediction. And then during the T cell epitope, I get a T cell the epitope, the peptide epitope of nearly 9 to 13 amino acid. So whatever I have 9 to 13 amino acid peptide isn't from the tool and the database. Okay. So that I will be taking it for molecular docking studies. So for docking it, I try to dock with MHC proteins or toll-like receptor proteins. Okay. So based on what exact immunity we want to get. Okay. So these are the protein target receptors we dock. Okay. I mean the peptides dock with the proteins and peptide protein complex, whatever we dock, we take further for simulation analysis, MD simulation analysis. So this is the workflow for the T cell epitope-based vaccine for Nipah virus. So this is one more example, like for SARS-CoV-2, if you want to work, okay, we have nearly 4 structural proteins and 16 non-structural proteins. In the 16 non-structural proteins, we can able to find the proteases, polymerases. So these are not considered as the major target, but these are considered as a major target for most of the viruses. But for the coronaviruses, okay, which is going to have spike glycoprotein on the surfaces, the spike glycoprotein is considered as the most important target. It comes under spike structural proteins. Okay. So one has to know about how to design even the vaccine, okay, how to select the target for a particular virus. If you see this multi-tope that is COVAX, I mentioned you, isn't it? That shows more than 99% efficacy. This is developed from nearly uh, 6 synthetic peptides. So one is universal. Okay, there is a helper T cell epitope, which is going to target the MHC2. Other than that, they are also used 5 specific cytotoxic lymphocyte and helper T cell epitopes that targets MHC1 and MHC2. You know, in USA, they find 5 important allele peoples. Okay, they've taken that. Okay, and they just designed these kind of a cytotoxic lymphocyte and helper T cell epitopes, and this they linked with a universal epitope like a universal primer for the DNA synthesis, DNA expansion. Similarly, here also they did a link with a universal peptide to target most of the SARS-CoV-2 strains. Okay, and then whatever 6 synthetic peptides they had, they linked with the receptor binding domain of structural protein of spike glycoprotein. Okay, receptor binding domain is a so on the they linked with this kind of fragments and whatever they linked. Okay, they also linked with CpG. Okay, and then they linked with an adjuvant. The adjuvant they used is aluminum phosphate. Okay, so this is the common adjuvant they use. Okay, so this is how a multi-tope vaccine looks like. So one has to design, one has to come up with the 5 specific peptides in this case. The remaining thing are the common groups attached. Okay. So for this, how to find these five peptides? So this we can learn from the next two sessions. COVAX, if you see, if it compares safety, efficacy, convenience, commercialization, however, for everything you can find this vaccine is excellent. Okay, this has shown excellent for the SARS-CoV-2 infection when compared to the monoclonal antibodies. You know, monoclonal antibodies are more precise, precisely targets the antigens, isn't it? But other than this, okay, even these vaccines, the co-vaccines, because it's developed from the peptides, okay, just like peptides are of course considered as a small proteins. Okay, so these small proteins are showing multi-targeting, or even it shows exact targeting on the binding sites. And of course, these are far better than the small molecule inhibitors. Now we can go to the T cell epitope. So epitopes, okay, are of two types. That is T cell and B cell. What do you mean by epitope? The epitope is the region, okay, is the region present uh, in the T cell. So if you see this, okay, um, I just don't show you. So actually, this can be considered as the T cell epitopic region. If this is the T cell epitopic region, okay, the one which is paratopic region is just quite opposite. Okay, so this is paratopic region. So T cell epitope, B cell epitope. And exactly if you see what is the epitope in this structure, okay, this line, okay, this I can draw it in another color. Yeah, so this is what the epitope they have shown. Okay, so this epitope region is large because it's coming outside also, isn't it? So for if you want to design a epitope for CD4 T cell, okay, which is going to target this is not MHC, but this is MHC2. CD4 the cytotoxic T cell targets MHC, no, yeah, cytotoxic T cell targets the MHC2 molecule of antigen presenting cell. And if you find a CD8 region in T cell, we call this as helper T cell. So this is actually cytotoxic T cell. So this is actually helper T cell. Okay, the helper T cell has CD8 antibody on the surface, which is going to interact with the MHC1 of antigen presenting cell. That okay, like how you find CD4 of TC is interacting with the MHC2. Here you find this. Now, if you see this kind of epitope, I mean, whatever epitope is going to bind with the paratopic region of your MHC. If you see this, okay, uh, the structure varies. Okay, the structure of MHC1, MHC2 varies based on the structure. This length will be a large here. Whatever peptide you design for the cytotoxic should be 13 amino acid. Actually, it is in the range nearly 12 to 18, something like that. But according to John Hopkins University, who majorly involved to develop vaccines, okay, at that time, okay, they proposed to bring the 13 amino acid figure for the MHC2 for this cytotoxic T cell. But here, if you see the helper T cell, they have suggested to go for 9 amino acid. Okay, here it is a shorter size. I think even in the figure you can able to understand. This is shorter length when compared to this larger length. So you can see subsequently in the next slides. Now also go to the B cell epitope. If you see the B cell epitope, okay, in T cell I told you there are two epitopes, cytotoxic and helper. How about in B cell? In B cell we have two types of epitopes. One is linear, the other is the conformational. So if you see the linear, okay, all the amino acids in the epitope is going to bind with the paratopic regions of the antibody. So all the amino acids in the epitope bind with the paratopic regions, but you find a continuous amino acids are there in the residue. Continuous amino acid residues are there in which interacts with the paratopic region. But if you see here in the conformational B cell epitope, or also called as discontinuous conformational B cell, also called as discontinuous B cell epitopes. Here you find the amino acids are discontinuous; they are not continuously present in the protein sequence. So if these amino acids are discontinuous, obviously you can find whatever epitopes binding with the antibody is perfectly bound when compared to here, because an amino acid is going inside. So when compared to this binding is more efficient. Okay, so sometimes you need a kind of a conformational or discontinuous B cell epitope for increasing the binding affinity. Okay, so either you can go for linear epitope, this is the first step, or you can go for conformational epitope for B cell. And if you see the receptors on T cell and B cell, these are specific for each and every antigen. You can't tell it can bind with all the kind of SARS-CoV-2, make it bind with a few strains, and it might not bind with a few strains, and what exactly you have to the design is for that you have to choose accordingly. Okay, so the B cell receptor looks like this, and T cell receptor looks like this. So whatever is shown is a kind of just one projection. If you see in the 3D, okay, if you the CD3, you find CD3 is bundled surrounding the alpha and beta regions. Okay, like in a cylinder, okay, the nearly four sets of CD3 molecules surround the T cell epitope. Okay, so it looks like that. Okay, let me explain about the peptide presentation MSC molecules. I've already explained you. Cytotoxic T cell try to bind, okay, with the MSC2 molecule receptor. Okay, but this alone won't give a kind of antigen presentation and process response. Okay, we also have to see that CD4, okay, from the T cell is interacting with the MSC2 molecule. So if and if you find these two, you can find that CD4 T cell gets activated. Okay, I think I have wrongly mentioned. Huh? The CD8 is cytotoxic T cell, but CD4 is the helper T cell. Okay, this is how you can find. CD4 is MHC2, then of course helper T cell. So this is the correct. Okay, and CD8 T cell is the cytotoxic T cell. If you see the helper T cell, that is CD4 T cell, okay, uh, is going to the epitopic region of the helper T cell is large, okay, around 13, but here it is small, around 9. Okay, so during execution of the tool, okay, you have to fix the size before developing. And if you see the dendritic cell markers, I just worked on dendritic cell, so I found nearly 162 markers. I tried to bind. I just worked on to bring a plant-based adjuvant for dendritic cell maturation. So I just tried the plant molecule, the binding of plant molecules with the 162 receptors, and I also found which is the my plant molecules bind with the what how many receptors. Okay, so I just tried with binding affinity of my plant compounds with the 162 receptor. So I'm just showing you. So if you find in a APC, that is an antigen presenting cells, or can be a dendritic cell, macrophage, or B cell. Okay. So for example, for APC, I showed you a dendritic cell. Okay, where you can able to find 162 or more than 162 receptors. I have taken significant receptors for checking for discovering a vaccine adjuvant. I tried the finding affinity of interactions of small molecules with this receptor, and also you can able to find the APC of that is an example of APC dendritic cell is to activate the T cell, it's going to have these kind of receptor interactions. Not only the T cell gets activated, so if you find an appropriate kind of interaction with DC and T cell, even DC gets activated. Okay, so if DC gets activated, T cell gets activated, the DC alone can even go and combat the cancer cells, and even T cell gets a kind of immunity. Okay, we have memory T cell also. These T cells also directly go and combat the cancer cells. Okay, so cytotoxic T cells directly go and combat. Helper T cells take a help with the cytokines for combating the cancer cells. So roles of MHC molecules. You have a lot of pathways. For MHC1, we have a pathway, and MHC2, we have a different pathway. So using these pathways, okay, if you see a cancer antigen is coming inside the cell, okay, these are actually degraded through the proteasome. So whatever proteins, okay, the proteins from the cancer, I mean the cells come inside, okay, these peptides go inside and try to bind with the MHC proteins, and these are transported to the Golgi and secretory vesicles and finally transported on the surface. Whatever antigens, cancer antigens are exposed on the surface in the form of an epitopes, try to bind with the T cell, okay, that is uh, T cell paratope. Now this whatever going to bind in the T cell is the paratopic region. This is the paratopic region, and the epitopic region is already there in the MSC molecules of APC. Okay, the paratopic region of T cell interacts with the ectopic region of the MHC, then obviously try to give the kind of an activation of the T cell. This T cell can go and combat the cancer cells. So structure of MHC molecules are explained, and not only you have MHC1 and MHC2, also have MHC3, which works on complement activation. Okay, the role of MHC cells, MHC1, MHC2. So all these things we'll be discussing in the next class. Okay, next session. So actually, in the next session, I basically teach you about the important database, the immunoinformatics database called IEDB, that is Immune Epitope Database. Okay, so here you have separate tools for T cell and B cell epitope. You have series of hyperlinks. You have to click inside, and you can use these tools for identifying the epitopes. And for getting the data in the databases, you have this information. Okay, you have so many data, so many epitopes are deposited in the database. Peptidic epitopes, 10 lakhs. Okay, non-peptide epitopes. Okay, so use a kind of a data mining from the tools already existing in the same website URL, IETP, Immune Epitope Database. Okay, that's it. So I've completed some basic information on immunoinformatics and the reverse vaccinology. So tomorrow we'll be discussing in detail about how to go for usage of tools for T cell epitope and B cell epitope vaccine design. Any doubts you can ask me. Actually, much immunology is involved, so kindly excuse I can't able to complete within the time. Okay, so one has to go through the immunology to learn, to acquire, to understand what exactly I want to infer. Okay, thank you. I think many of them don't have any doubts. So thank you for your patience and the interaction.